Now we are talking about the NK cells, that are natural killer cells, that are natural cells, that are basically they are originated from the lymphatic progenitor cells. Okay, now NK cells, they do not perform phagocytosis process. Phagocytosis process do not occur in NK cells. Okay, like they will not engulf any type of bacteria. Okay, like they will help, they basically help infected cells. We'll discuss that how they help infected cells. They detect either viral or bacterial cell. They will detect that viral, either the cell either that is the viral infected cell or the bacterial infected cell, or sometimes they detect the cancerous cells as well. Okay, basically if there is one thing, that if MHC1 is present on any cell, MHC1 is present on any cell, it means that cell is infected. Like, okay, now MHC1, like NK cells will detect that either there are many MHC1 are present, how many MHC1 are present on that infected cell. Now, like when, for example, this is a cell, body cell, and this is a virus. Now when virus enters into that cell will get infected. As I said, when the cell will get infected, MHC1 will be present here. MHC1 will be present because when virus will, for example, if the pathogen is virus, then virus will enter into the body cell, and then the cell will got infected. Now what will happen, that that infected cell, the cell will got infected, and that infected cell will release interferon. Okay, that infected cell will release interferon, and basically interferon will activate NK cells. NK cells are inactive, and as infected cell will like activate release interferon, then interferons will activate the NK cells. As MHC1 is present on the cell, body cell, so that will on that infected cell, then MHC1 and virus that enters into the cell, they both will fight together. When they will fight, MHC1 will take a one part of the virus on its surface. It will take one part of the virus on its surface. When the NK cells activated by the interferons, these NK cells will move to MHC1, and then MHC1 will read that then MHC when MHC NK cells will read that MHC1, that what enters into the body, and as a virus is on its surface, on the MHC1 surface, NK cell will read that. Now NK cell will produce perforins. Now what are perforins? Perforins that will work as a perforation, work for the perforation. Perforation means holes. Like they will do holes in the membrane of the cell. That perforin will be released by the NK cells, and then perforins will do perforations, means there will be the holes in the membrane of the cell. Then again, as their holes are done, then NK cells will again release an enzyme that is known as granzyme. What granzyme will do now, what NK cells will do, NK cells will like insert that granzyme into these holes. Now NK cells will, as NK cell has released the granzyme, NK cell will like insert these enzymes into the cell by these perforations. By these perforations, granzyme will insert into that like cell in the body cell, that infected cell. Then NK cells will detect that how many MHC1 is present there. If there are many MHC1, that it means the cell is infected. But if there is only like not more MHC1 is present, it means the cell got infected. But if there are many MHC1 are present, so it means that the cell is not infected. Okay, so NK cells work for the detection of the cell, that either the cell is infected or the